Good morning, everyone, and welcome to today's presentation. I'm delighted to have you here. Today, we'll be discussing the management of samples for chemical and microbiological analysis. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the next few minutes, I'll guide you through the key aspects of handling and managing samples to ensure accurate and reliable test results. We'll cover best practices, important considerations, and some practical tips to make your sample management process more effective. Let's get started. Today, I want to walk you through the main objective of this slide. Our goal is to clearly outline the standardized procedures we follow when handling, processing, testing, and reporting samples of formulations and APIs. This applies to both chemical and microbiological analyses in our QC laboratory. Ensuring consistency and compliance is key here, so we focus on maintaining traceability and data integrity at every step. Let's also clarify some important definitions. A sample is simply a portion of the API or formulation that we analyze. Chain of custody refers to the documented tracking of the sample's movement through each stage, which is essential for accountability and traceability. Holding time is the maximum allowable period before the sample must be analyzed to ensure the results remain valid and reliable. This procedure covers a wide scope, anything from raw materials and intermediates to the finished API and final formulations. It's designed to ensure that all samples are handled consistently, safeguarding the quality and integrity of our testing process. This slide sets the foundation for understanding how we maintain rigorous control in our laboratory operations, aligning with our overall commitment to quality and compliance. When we talk about key guidelines and standards, we're really emphasizing the importance of following established practices to ensure quality and consistency. First, there's good laboratory practices, or GLP, which set the framework for conducting reliable and reproducible lab work. Then, we have good manufacturing practices, or GMP, which are crucial for ensuring that products are consistently produced and controlled according to quality standards. These GMP guidelines are recognized by organizations like WHO, US FDA, and EMA, highlighting their global acceptance. Next, the ICH-Q2, R1 guideline focuses on the validation of analytical procedures, making sure our testing methods are accurate, precise, and reliable. Validation is a cornerstone for quality control in any analytical work. Then, we have ISO IEC 17025, which defines the competence requirements for testing and calibration laboratories, ensuring that labs meet international standards for technical capability. Finally, we can't forget pharmacopoeial requirements, like those from USB, IP, and BP. These provide the official standards for drug substances and products, guiding quality and safety. Overall, these guidelines and standards form the backbone of our quality assurance framework, ensuring that every step from lab to manufacturing meets rigorous requirements, thereby safeguarding public health and maintaining trust in our products. Now, let's take a closer look at the different sample types and the compliance requirements we need to keep in mind. First, we have the API, or active pharmaceutical ingredient. This is the core component that provides the therapeutic effect in the medication. Next, we have finished formulations, which include tablets, capsules, liquids, and creams. These are the final products ready for distribution and use. Lastly, we have in-process samples, which are taken during the manufacturing process to ensure everything is proceeding correctly. Moving on to compliance must-haves, there are a few key points to emphasize. Proper labeling and documentation are absolutely essential. They ensure traceability and help us meet regulatory standards. Equally important is conformance to stability and storage conditions, meaning we need to verify that samples are stored correctly to maintain their integrity and efficacy. All of these elements tie back to our overall goal of ensuring quality, safety, and regulatory compliance throughout the manufacturing and testing process. Keeping these sample types and compliance essentials in mind helps us maintain the high standards expected in our industry. Let's walk through the key steps for receiving a sample from production or sampling. First, it's crucial to verify the sample transfer documentation, such as batch records and the sampling slip. This helps ensure that the sample is properly documented and traceable. Next, we need to inspect the sample itself for integrity, check the seal, look for any tampering or damage. This step is vital to confirm the sample hasn't been compromised. Then, we confirm the quantity and sample type to make sure it matches what was requested. Finally, we record the date and time of receipt, along with the responsible personnel's name. This completes the documentation process and keeps everything transparent and accountable. These steps are fundamental to maintaining quality and compliance throughout the sampling process. Now, let's talk about the sample registration process. First, it's essential to assign a unique sample ID to each sample. This helps us easily track and differentiate samples throughout the entire process. Once we've assigned the ID, the next step is to enter the sample details into our sample register or laboratory information management system, often called LIMS. This ensures all information is stored securely and is accessible for future reference. When labeling the sample, there are some critical pieces of information that must be included on the label itself. First, the sample ID, which we've already assigned, and the product name, so we know exactly what the sample is. It's also important to include the batch or lot number, which helps with traceability in case of any issues. Don't forget to add the owner or requester's information. This tells us who requested the analysis or who is responsible for the sample. The label should also specify the type of analysis to be performed, whether it's chemical or microbiological, so everyone knows the scope of testing. Finally, the label must include the receipt date and time, as well as the due date for the analysis. This helps us keep track of turnaround times and ensures timely results. By following these steps, we make sure our sample registration process is thorough, accurate, and aligned with our overall quality management goals. Now, let's talk about storage and preservation. It's essential to store samples according to the specific conditions outlined in the guidelines. This means using controlled room temperature, refrigeration, or freezing, depending on the sample type. Proper storage helps maintain the integrity of the samples over time. It's also important to protect samples from light and humidity when necessary, as these factors can degrade certain specimens. Remember to keep chemical samples separate from microbiology samples to avoid cross-contamination and ensure safety. Regular monitoring of environmental conditions is crucial too. This means checking storage temperatures and humidity levels consistently to make sure everything stays within the specified ranges. By following these storage and preservation guidelines, we're ensuring the quality and reliability of our samples, which ultimately supports the accuracy of our results and the success of our overall research or testing process. Let's talk about how we distribute samples to analysts in a controlled and secure way. First, we issue the samples through a distribution log or LIMS system, which helps us keep track of who received what and when. It's important that each analyst signs off and timestamps when they receive the sample, ensuring accountability and traceability. We also provide clear instructions on the priority of the samples and the expected completion times, so everyone knows how to handle them efficiently. Now, to minimize cross-contamination, we emphasize the use of appropriate protective equipment and aseptic handling techniques. This means wearing gloves, masks, or other protective gear as needed, and handling samples in a way that prevents any transfer of contaminants. Maintaining this level of care is crucial to ensure the integrity of our samples and the accuracy of our results. Overall, these steps are vital parts of our quality assurance process, helping us maintain sample integrity, ensure data reliability, and uphold the highest standards in our work. Let's take a closer look at the microbiological analysis workflow. First, key activities include preparing the media and confirming sterility to ensure no contamination. Then, we move on to inoculating samples appropriately and incubating them under the right conditions to promote microbial growth. We also perform enumeration, which involves testing for total plate count, yeast and mold, and specific pathogens, depending on the requirements. Environmental controls and negative or positive controls are crucial to validate that our testing environment is reliable and that our methods are working as expected. Finally, detailed documentation of each step ensures traceability and compliance. Now, shifting to the chemical analysis workflow, the first step is to check the validity and suitability of the method we're about to use. This is essential to guarantee accurate results. We also need to calibrate our equipment and verify system suitability before starting analysis. Sample preparation involves weighing and diluting samples properly to get representative and consistent results. Then we perform the analytical execution, whether it's an assay, impurity testing, dissolution, or content uniformity, to obtain the necessary data. Throughout this process, recording raw data and chromatograms meticulously is vital for data integrity and future reference. Together, these workflows form the backbone of our analytical processes, ensuring that our results are reliable, reproducible, and compliant with quality standards. Now, let's look at data review and quality assurance. When reviewing data, it's essential to check for completeness, traceability, and validity. This means ensuring all data is fully captured, can be traced back to its source, and is accurate. We also review calculations and chromatographic integrations to confirm they were done correctly. Cross-checking the data against specifications helps us verify that everything meets the required standards. Additionally, it's important to have the analyst and reviewer sign and date the data, which maintains accountability and traceability. On the QA side, oversight involves making sure we follow standard operating procedures, or SOPs, at all times. We also monitor the stability of standards and ensure our instruments are performing optimally. This comprehensive approach helps us maintain the integrity and quality of our data throughout the process, which is vital for reliable results and overall confidence in our work. Now, let's talk about report generation and approval. When we prepare a report, it must include several key elements. First, it should have sample details and identification to clearly specify what was tested. Next, we need to include test results and compare them against the specification limits to see if they meet our standards. It's also important to attach raw data, like chromatograms and counts, to provide transparency and traceability. If there are any nonconformances, these should be documented thoroughly, along with the rationale behind them. Additionally, the report must have authorized signatures and dates to verify accountability. Remember, final QA approval is a crucial step before we release the product or decide to reject it. This approval acts as a gatekeeper, ensuring everything has been checked properly. Overall, this process helps maintain the integrity and reliability of our testing, aligning with our goal of consistent quality assurance. Let's talk about sample archival and disposal. First, regarding retention policy, it's crucial that we store tested samples according to the regulatory retention timelines. This means keeping them long enough to meet legal and quality standards. We also need to maintain the integrity of those samples throughout that period, ensuring they remain unaltered until disposal. When it comes to disposal, safety and environmental responsibility are key. Chemical waste should be disposed of following environmental and safety guidelines to prevent any harm. Microbiology waste, on the other hand, must be sterilized or autoclaved before disposal to eliminate any potential biohazards. And finally, it's important to keep proper records of all disposal authorizations. This ensures traceability and compliance with our regulatory obligations. Connecting this to our overall theme, proper sample management and disposal are vital components of maintaining quality, safety, and regulatory adherence in our operations. Let's talk about some critical GLP and compliance practices that are essential for maintaining integrity in our work. First, it's absolutely vital to follow validated methods strictly. This ensures our results are reliable and reproducible. Next, making sure that instrument calibration is current is key. Out-of-date calibrations can lead to inaccurate data. Maintaining a clear chain of custody and accurate documentation helps us track samples and procedures, which is crucial for audit trails. Regular internal audits and ongoing training are also important. They help us identify areas for improvement and keep everyone up to speed on best practices. Lastly, if any deviations or issues arise, immediate reporting and corrective actions are essential to prevent problems from escalating. These practices, when consistently applied, form the backbone of a robust compliance program and ensure our work meets the highest standards. For further support, please don't hesitate to reach out. If you have any questions or need additional assistance, we're here to help. Feel free to contact us through the provided channels. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Thank you once again.